Hi everyone, thanks a lot for coming today. This talk is part of a Young Scholar Seminar series. And first of all, I would like to thank Yunus Emre Enstitüsü for having me today. So today I am going to talk about some water pathogens, and specifically the talk title will be What's Living in Your Shower. I am Özge Yetiş. I am a postgraduate research scientist in University College London Hospitals Environmental Research Laboratory. I am going to talk mainly about my PhD studies today, which is investigating hospital water contamination and its risk for infection. So to start with a bit background about me, I did my PhD in UCL, Division of Infection and Immunity, and I graduated in 2023. Before, I did my bachelor's in Istanbul Technical University, Chemical Engineering and Molecular Biology and Genetics, a double major. So to give you a bit outline about our presentation, first of all, we are going to talk about water pathogens, specifically Pseudomonas aeruginosa. We will talk about hospital-acquired infections, and then we will move on to showers and talk about current water sampling practices. And I will talk about the first two studies of my PhD thesis. The first one is longitudinal sampling for Pseudomonas aeruginosa, and the second one is focusing on shower filters, particularly membrane and hollow-fibre filters. Then we are going to talk about screening studies and put the clinical importance of all these environmental findings that we discussed, and we'll focus on whole genome sequencing. So first of all, water pathogens, they are parasites, mainly viruses and bacteria, that can be found in various water sources like lakes, sea, our tap water, and we can get it through eating contaminated food or through recreational activities, taking a shower, like anything really. So you see the site of infection, maybe it's a bit small to see, but mainly this is the mucous membrane of our nose, our respiratory system, gastrointestinal system, urinary system, and skin, all around our body. First of all, the smallest waterborne parasites are viruses. They are very small, typically between 20 to 300 nanometers. Here are some examples: adenoviruses, enteroviruses, and noroviruses, and also rotavirus are one of the most important ones. The diseases they are causing are mainly gastroenteritis and respiratory infections. Its infectivity is really high, and we can get it through ingestion or inhalation. And here you can see a diagram of a virion. Secondly, we have waterborne bacteria. There are quite a few examples of this. One of the most important ones is Vibrio cholera. This bacteria group has more than 200 serovarieties, and it produces cholera toxin, and this acts as ionic flux across intestinal mucosa, causing severe diarrhea, which is causing to lose lots of water. Other important bacteria are Salmonella enterica and typhi, and Shigella, Escherichia, Burkholderia, Campylobacter, and Legionella and Pseudomonas that our lab is focusing on. Here at the beginning of my slide, I put an interactive question for you. So this is a Slido question. If you can scan the QR code and try to guess the answer of this, please. So as I mentioned about Vibrio cholera, that is causing diarrhea. So I would like you to answer how much water a person can lose if they have the infection. Sorry, I think as you are, as the participants are coming. Yep, we have 6, 7. I will give a few more seconds. So most of you guessed 5 to 8 liters per day, which is understandable, but the correct answer was 10 to 15 liters actually. So it can be really severe if you think about that water loss in a day. So continuing on other important waterborne bacteria, we have Campylobacter, E. coli, these we already discussed, Shigella and Vibrio cholera. For the purpose of today's talk, we are going to talk about Pseudomonas aeruginosa. But before that, I would like to introduce hospital-acquired infections. These are the infections acquired by hospital visits or during our stays in the hospital, can be acquired by the patients, visitors, or healthcare personnel. So the definition says if the infection starts in the first 48 hours of admission or visiting a healthcare facility, that is a hospital-acquired infection. It can be surgical, urinary, or respiratory infection. One of the main bacteria causing hospital-acquired infections, or nosocomial infections in other words, are Acinetobacter, Klebsiella, Clostridium difficile, E. coli, and Pseudomonas aeruginosa. Infection control is very important to combat with this. So here you see the importance of hand wash. We usually underestimate it, but it's very important to wash hands frequently, at least for 20 seconds. So moving on to my bacteria, which is Pseudomonas aeruginosa, it's one of the leading causes of hospital-acquired infections, mainly targeting immune-suppressed patients, meaning the patients who has cancer or chemotherapy. Because this is an opportunistic pathogen, it doesn't necessarily infect an healthy individual, but it rather waits for the immune system to be suppressed to attack. So here you can see the capsule, flagella, and its pigments, which are taking part in its in infection strategies. It is also important because it has antimicrobial resistance properties, both innate and adaptive ones. So due to membrane impermeability, it's already immune to some of the antibiotics, and it can gain resistance very quickly. And here you can see on a culture plate how it looks like. Maybe you might recall this from the background of my presentation invitation. So here is a diagram showing its biofilm properties, because once it attaches a surface, it secretes some extracellular material, and then it attaches, and then it disperses to the other places, and this process starts all over again. And once it does that, it gets very difficult to eradicate. So hospital water systems are usually colonized by Pseudomonas aeruginosa. This is an aerobic pathogen, so it chooses distal parts of the plumbing system and grows, proliferates there. Here in the UK, Health Technical Memoranda sets up some guidelines for sampling water for Pseudomonas aeruginosa. Especially this part focuses on Pseudomonas in augmented care units. If you are not familiar with this term, this is used for the wards that cancer and immune-suppressed patients stay. Here is another question for you. So in the UK, how often do you think water should be checked, according to the guidance, should be checked for Pseudomonas aeruginosa? Nine of you already attended that one. Great, so most of you think it should be checked every day, which I really wish, but the correct answer is every 6 months. And we are going to discuss if it is frequent enough. So this is the diagram given in the guidelines. So the water sample, after it's collected, if Pseudomonas is not detected, nothing we need to do. If there is 1 to 10 colony forming units per 100 milliliter, then we need to retest because there could be some external contamination maybe. But if it is more than 10 CFU, then first risk assessment should be done, and then a remedial action such as a disinfection or the tap change, shower change should be done. And after that, sorry about that, it should be retested after 3 days, and then after 2 weeks and another 4 weeks. So all of these days we are checking if the contamination is still there. If it is still there, it goes back to remediation. So that means our disinfection or whatever we did didn't work. So this process starts all over again. But let's say if an outlet gets cleared by this remedial action, or if it didn't have any from the start, that means according to the current guidelines for 6 months we don't need to do anything, but we are going to discuss about it. So shower contamination, these are real photos from hospital wards. First of all, if the shower hose is very long, this is a 1.2 meter long hose, it can touch the floor, and you can see like they just tried around so that it wouldn't touch the floor, but this is not a good idea. Here is also a long hose, which you might think like, what's the problem in this one? It looks okay, but it's actually, as water stagnates in this U-loop, that also promotes bacterial growth. For this reason, the hospital changed the hose length to 0.8 meters. So it's actually the idea of it. It first started as it will be mounted on this level, but then the recommendation was to leave the shower head so that all the water will just, and it wouldn't touch the floor, and all the water will flow away, so there wouldn't be any stagnated water either. So what's living in your shower? We opened up some of the shower heads. Again, this is a real world example, and you can see is slimy material, like this is a polymicrobial community living here, and this is the shower hose. You can see the same bits here, and trying to analyze which bacteria is there, we took a piece from the shower head, and also this was a bit easier to cut, and then we washed it several times, and you can see the resulting plates after the first wash. So here you can't see clearly because the bacterial count is very high, so we tried to dilute it 10 to -1, -2 to be able to count the bacteria, but the levels were very high, and this was actually after the biofilm removal protocol. So we tried to disrupt whatever is in here and then tried to wash away, but still the bacteria Pseudomonas persisted, so that means that already moved to biofilm state. So from this picture, all of these species were identified, so it wasn't only Pseudomonas species. We found Aeromonas, Citrobacter, Achromobacter. Stenotrophomonas maltophylla is another important hospital pathogen because it has multi-drug resistance properties, and also Enterobacter. So here, from here I move on to my first study, which was a longitudinal sampling study. We chose 2 wards from the hospital, and this is when I started my PhD. We sampled those showers and drains for 7 months with the frequency of every week or every 2 weeks. So here we collected water and we filter concentrate on a membrane and then plate it, and here you see a heavy growth because Pseudomonas usually grows yellow or green. And from the drains we just sticked some cotton swabs and then directly streaked on an appropriate plate. And from here, the bacteria we found, we need to do some confirmation tests. For Pseudomonas, we do oxidase and casein hydrolysis test. So this is a casein plate. This is milk cetrimide. So you see this 1A 4A and 4B, they hydrolyzed the milk, whereas the other ones, even if they grew, they didn't hydrolyze the milk. And most of the Pseudomonas have fluorescence properties, and you can actually see me here trying to take a picture. But it doesn't mean that is like either a positive or a negative, but we know most of them have this property, so we make a note about it. But this is actually like a culture-based method, but now we moved on to MALDI-TOF as well. This is a mass spectroscopy. So from all of these isolates, I also did like a confirmation with the MALDI. So let's see the results of these 14 sampling weeks. Many interventions happened during this study, because when you see an outlet like this, you can't really leave it according to the guidelines we just talked about. So here, this particular example, this outlet was filtered at the beginning, so the water was actually contaminated, but because of the filter, it wasn't coming out. Then the hospital changed showers to antimicrobial products. Actually, antimicrobial in quotation marks, because you can see as the bacteria grows here, it's either not efficient or maybe the concentration is not high enough to kill the bacteria. So here you see, for example, on week 4, we started seeing colonies, and when it moved to week 5, it was already greater than 300 CFU, and it carried on like this until the final intervention, which made it clear. But I am going to show you like the results all together. This is a busy slide, but I will just show some examples. So through 7 weeks of 14 sampling points, the green ones show the shower water contamination, and the orange ones the drain contamination. So if you see, let's pick an example. Most of the cases, even though the outlet was clear, the contamination started after some point, and these are interventions, but regardless of the intervention, it just carried on and stayed highly contaminated. The drains is a bit more difficult scenario, because in addition to Pseudomonas, many other bacteria also grow, because imagine drain environment, like lots of things are put through, also disinfectants, also like skin. So it's like a very rich environment for the bacteria to grow. So at times it was a bit difficult to isolate Pseudomonas aeruginosa, because other bacteria could be overgrowing or killing Pseudomonas, but we still saw high rates in both shower heads and drains. And if you are interested in the details of this study, this is published in Journal of Medical Microbiology, so feel free to have a look. And here we have some regression models also trying to model the contamination rate of showers and drains. So as I show in one of the previous slides, like some of the outlets had filters. Now we are going to talk a bit more detail about those ones. So the conventional ones were membrane filters. So the pore size is between 0.1 to 0.45 micrometers. So this is smaller than our bacteria size. So even if the outlet is contaminated, that means at the point of use, patients will have safe water if the filters are working properly. But one of the difficulty with this one is that it needs to have adequate shower pressure, because otherwise it's not going to give enough flow rate. And we noticed when I went to the wards to sample those showers, we noticed patients tend to take out the shower heads, because you can think about yourself as well. Like if water is not coming, the first thing you think of doing is something wrong with the head, so let me take it off. But it was actually protecting them, so putting themselves at high risk with the contaminated water. So because of these issues with the conventional filters, now there is a new technology with hollow-fibre filters. So this is a cross-section of what is inside the shower head. These are really fine 0.2 to 0.1 to 2 micrometers pore-sized filters, hollow fibers. Again, the same idea is to filter out the outlet. This has increased membrane life and more structural strength. But we did a study on 25 of these showers because we noticed the infection rates were not really dropping. And here is another paper, you can check the details. Because out of 25 of these hollow fiber shower filters, is actually 8 of them had Pseudomonas in the outlet. So there could be many reasons. It might not be that the filters are not working. It could as well be an external contamination. But here, I think for this purpose, the cause is not that important. It is very important that patients were unfortunately exposed to this bacteria. And the details are here. This is published in Journal of Hospital Infection. So up to here, we all talked about like always the environmental Pseudomonas, but what is the clinical importance of it? What is the problem with the patients? For this one, we did a screening study because now we demonstrated that the bacteria is there, but also we need to know if the patients are already coming as carriers or they already have infections. So that's why we chose one ward for this study, which was actually the same ward as my first study. And 155 patients were involved, 606 samples were collected, they were groin and rectal swabs. So the prevalence at admission, the patients coming already with the infection, was low, 1.3%. And during the course of the study, 4 patients who were negative at the admittance actually acquired the infection during their course of stay. So again, one can argue, is this also a low rate, like 2.6%? But if you think like even one patient is very important, and also, again, this is a published study. If you check the study period, this is right the beginning and the first wave of COVID. So we needed to cut the study a bit short because most of the wards were turned into COVID wards, so that wasn't very feasible to continue. And also one other problem with these carriage could be that because hematology patients are involved and it is difficult to take groin and rectal swabs from those patients because of chemotherapy, the perineal area can be easily damaged. So patients can as well refuse this, and they have the right to do so. So maybe the levels could be lower than expected. So this screening study showed us the clinical importance of acquiring it during an hospital stay. And now I want to show you an example of the antimicrobial resistance properties of Pseudomonas aeruginosa, because right at the first slide I said this is one of the biggest concerns. So here, let me open. I am just showing you like how we do it in the laboratory. This is a disk diffusion assay. So we first filter sterilize our forceps, and these are the discs that antibiotics are already impregnated, and then these plates are they already have the bacteria, so they will grow in the presence of the antibiotic discs for us to check the susceptibility patterns. And here, this is a bit more automated system that we don't have to put it one by one, because the plunger puts 6 discs per plate. And these plates take at most 6 discs, and I am going to show you the resulting plate. Okay, good. Let me stop this. Okay. So why is this important? After we put the discs, we incubate the plates, and here you see the inhibition zones, and here the green area is where bacteria grew. So these inhibition zones mean that the antibiotic works effectively because the bacteria couldn't grow with the presence of the antibiotics, and here you see 12 different antibiotics belonging to five classes, I think. Fully susceptible pattern, this was a water isolate. And here on the second example, you see actually bacteria grew regardless of the antibiotic. So they were resistant, and this one was a multi-drug resistant bacteremia, is a blood infection isolate. And even this is a very small zone, and we have guidelines telling us like which measurement will be resistant, intermediate, and susceptible. For this particular antibiotics, it was all resistant, so this is a multi-drug resistant strain. So here the question is, it might not feel that important if all the environmental isolates are susceptible, but given that they can easily gain resistance with the presence of all disinfectants, antibiotics, and everything we use in the hospital, this is a big concern, and this is just one example I am showing. We have resistance patterns in environmental strains also. But to be sure about this, to be sure why this water bacteria important, we have to do molecular typing. So here, unfortunately, I won't show our results because they are still in progress. We haven't published them yet. But many studies showed, for example, this is a good one from Pseudomonas aeruginosa in intensive care units done here in the UK. They showed relatedness of water isolates and clinical isolates. And the acquisition is still, the question is still remaining, because the transmission could be environment to patient, patient to environment, or patient to patient. Even though you show the relatedness, this question is not clear yet, because we need to know the evolutionary history of the isolates, which isolate came first. It's not enough to know which one we isolated first, because we can only do so much. Like maybe I am sampling today, but this isolate, where did it start? I am not sure. So this needs some more additional work, and some people developed algorithms to estimate these rates, which we are trying to use. Before, pulsed field gel electrophoresis, variable number tandem repeats, and multi-locus sequence typing were used, which were also very important. But now, with the development of whole genome sequencing, we can have more robust results. And it's not only showing the relatedness, actually. We can also focus on antimicrobial resistance properties, which biofilm genes are there, or any other property that we want to check. And we can compare it with the results I showed you, because this will give us the genotypic answers. And phenotypically, we do our tests in the laboratory. So in conclusion, I think we know that waterborne pathogens are really important, and they are a big risk for human health. Pseudomonas aeruginosa being one of the leading causes of hospital-acquired infections, my sampling results and many other studies showed that hospital water, in our case, especially hospital showers, were the main reservoir. The disinfection and intervention studies are often not effective because of the table I just showed you, even though the contamination is eradicated, it comes back within days, maybe weeks. And molecular studies, they are showing relatedness between these patient and environmental strains of Pseudomonas, so that's why we need to keep our focus on the environment and disinfection strategies. And in acknowledgements, I would like to thank my primary PhD supervisor, Peter Wilson, and my secondary PhD supervisor, Sean O'Malley, and all the environmental research laboratory team, one of which is with us today. And I took my PhD funding from Turkey Ministry of National Education. I would like to thank them for giving me the opportunity. Some part of the sequencing work, which is still ongoing, is funded by seed funding from UCL Precision AMR grant. And I would like to thank you all for coming and listening. Happy to take your questions. Thank you very much. Yeah, are there any questions or comments?
I'm wondering, during the testing of the...
You have the microphone up there.
I'm actually wondering what's happening to the water facilities. I mean, patients are allowed to use them during the testing time. Let's say if they found like 10 CFU per 100 ml.
Yeah.
What's happening to the water facilities?
Yeah, very good question. Is actually they are not supposed to use them because it's not safe to use. It's also important the time of the sampling because it really changes during the day. So it will change if you take the sample early in the morning or after the patient's use. But usually what happens before the proper remedial action is done, those water outlets are taken out of use so that until it gets cleared, no one should use them. Any other questions? Yeah, Jill.
Thank you for the presentation. It's a very lovely presentation.
Okay.
So I was wondering if you had opportunity to collect samples from ICUs or theaters, because you know, in the ICUs also, if the water reservoir is the same with the boards, they may affect on the, I don't know, any patient. So so basically in the, we are working on Klebsiella or other stuff, but it's in the African countries, and they have a big problem with the Klebsiella infection in the neonatal ICUs. So, and they assume this might be one of the reason. So have you had any opportunity to look at?
Yeah, very good question. Yes, we did, but the difference in our hospital is that, you know, ICU settings don't have many showers, maybe any showers, because the patients would be too sick to take a shower, so they will only have taps. In our hospital, the problem is mainly with the showers. The taps in general, they are clear. So comes back to your question, like, because the water system is connected, yes, it is a good point. Because of that reason, at the beginning we checked the mains of the hospital, the main water tanks, because if it's the problem is already there, you are right, there will be everywhere. But in this, for example, even though this is a very bad table, but you can see, yes, most of the outlets are contaminated, but not all. So that means it's not coming from the mains, and we don't have a big issue with the taps. So that's why we can't really relate with ICU taps. This is for our hospital, but many other studies, as you said, they focus on ICUs. And when they have a problem with the taps, they can also, as you said, check Klebsiella or E. coli. But I think every water system has its own issue. For us, it's Pseudomonas aeruginosa. I think back in Turkey, Acinetobacter is one of the leading ones, so it might change. Thank you. Yes, please.
Thank you, and thank you very much for the presentation. Very, very interesting, fascinating. My question's about the filters.
Yeah.
So two elements to it really, I guess. One, most manufacturers recommend that those filters are changed periodically, and I think it changes from manufacturer to manufacturer. Some will say every 30 days and some will be longer than that. And I'm just wondering, because your study goes beyond the life of one of those filters, for example, whether or not you found that maybe filters that had exceeded that timescale might have been still giving you data where you'd expect an in-date filter might not. I wonder if you could deal with that bit first.
Yeah. Yeah, that's a good question. And here in this study, it wasn't the case for all of the showers. So even in dated showers within their time span, they had problems. But it is true that some of them exceeded their lifespan. So we actually took them out from our total number and tried to give like a good representation of, because we can't really, if it's already expired, we can't expect them to work functionally. But still the undated ones were a bit problematic in our sampling.
Yeah, okay, that's really interesting, because I'm aware that I'm not talking about your hospital or its trust, but I'm aware of others where a delay in timely replacement of those is going to have quite catastrophic, in some cases, consequences, because it's just not recognised that those filters will then start letting bacteria through. So that's that bit. The other question, it's kind of a little bit more, a little less directly aligned to this, which is that where you get, and you touched on a little bit earlier about where the source of the bacteria may be, and if it's in the main system, there's colleagues talking about the potential to impact ICU or other critical areas of the hospital. Whether or not it's been factored in that those remedial actions that are taken to reduce the risk at the shower. Obviously, you've got the problem with aerosol-based bacteria from a shower that you necessarily don't get from a tap. So even if it is present in a tap, you're much more likely to be at risk from it if it's from a shower. But if the actual source is not from that shower head, but somewhere further back in the system, even if it's not attacking the taps, for example, in dead legs, which you get where a ward might be decommissioned and then water's going into there, stagnating there because nobody's flushing the taps, but then it's being fed back into the shower system. I'm just wondering whether or not your study factored in those kind of independent factors that might have been present elsewhere in the piping system besides at the shower head. That's a really sophisticated long question. I'm sorry about that.
It made sense to me. Don't worry. Yeah. Thank you very much for your question.
Sorry.
Going back to the first one about the lifespan of the filters, I just wanted to add, usually we couldn't really leave them longer because they get blocked because of the lime scale. So then we turn back to either the estates or the manufacturers that maybe even replaced before their time of expiry. Coming to the second one, yes, I was going to put some slides of those ones because, as you said, we couldn't solve the problem by just changing the shower heads or the hoses. Then there is the question like, where is the contamination? And we went behind the walls as well. One study we did behind the wall, there is this TMVs, they are called thermostatic mixer valves. So we used parasite solution trying to disinfect them because they were also contaminated behind the wall. But this was kind of like a trial, and it was only affected for 1 out of 5 showers, and the contamination came back quickly. The second part with the flushing, we have a scheme which is like routinely done by hospitals engineers. So there didn't seem a problem with that one. The other question we are trying to answer, like how connected is this plumbing system that is affecting some of the showers, some of the outlets, but not all. So probably something to do with the dead legs, that I am sure. But currently we are finding it a bit difficult to access the actual plumbing plan of the hospital because I think they don't maybe want to share with us that one. But if we can find out, I think we can actually put the dots and then maybe have like an idea about how is it moving across. Yeah, I hope it answered your question.
Well, thanks, yeah.
Thank you.
I know I'm not on the microphone for this bit, but that's probably best I'm off the record. It's probably the reason they haven't shared the piping plan with you, because they don't have one. That's probably more likely the reason, or that it's not accurate and up to date.
That we thought, but I found it very interesting if they don't have one, because how was it planned at the beginning and we don't have it anymore? Yeah.
But things change, you know. Wards move around, taps move.
Exactly, and maybe they are not recorded, they are not updated. Yeah, could be. Thank you very much. Any other questions or comments? Yes, Kürşat.
Thank you for presentation. It can be a stupid question, but I don't know. I visited a hospital 2 times in UK, and I observed 2 different shower orientation. One of them include the toilet and shower in the same place, and other is separated toilet and shower. I wonder, the toilet can be affected to increase the infectious bacteria.
You mean the same place? You mean the same room, right? Yeah. Could be, yes. I think it's a valid point, because we know the splashes can travel like really long distances, but I wouldn't think that a splash from the toilet will go to the shower and then start a contamination straight from there. And also the bacteria found in the toilets are a bit different than the rest of the water system. So when you see it, you know it's usually like an external contamination, and these outlets are also wiped by disinfectants, which I think would be effective of clearing them. But I don't think it can travel across the water system and then create a problem. Where is? Yes, Cenk.
My question's from point of patient's view, because in the beginning you mentioned that these bacteria wait docile until the conditions met. When the patient's immune suppressive, then they act, right?
Yeah.
Could it be the other way around? Maybe the bacteria is tweaking the immune system of the patient, because there are some bacteria known to tweak their immune system, like Enterococcus faecalis. They can be micropinocytosis into the cell, macrophages, and treat macrophages to their benefit.
Yeah, a very good point. I think we can't say clearly this way or the other. I think both could be the answer. Like just like a textbook knowledge that they will infect immune suppressed patients. Why I specified that part was like they wouldn't necessarily infect us, let's say, the healthy individuals, that why the immune suppressed ones are a higher risk. But could it be the other way around? Could be, because I didn't go to that much in detail, but also for Pseudomonas, it's possible like they can translocate easily in the body. So maybe you already have it, but if it triggers some sort of activity, I am not really sure about that. I don't want to say anything wrong, but it's a good point, I think. Thank you.
Why this study is focusing on hospital water? So can we have this bacteria in our home water as well? So is it safe to drink normal tap water?
Yeah, that's a bit related to for Pseudomonas being an opportunistic pathogen. I don't want to scare you, but I think it's highly likely that you have it at home already, but it's not going to affect you, because you might also have it in, like, let's say, contact lens solution. Like, is okay to have it. And why are we focusing on hospital water? Because the target people, like the patient group, is there, so they are vulnerable to this infection. Whereas, for example, sampling all these contaminated outlets, I think I am already exposed to most of it, which didn't affect. So that's the reason, like they are staying there and then they already have lots of complications. Imagine a cancer patient taking chemotherapy open to many different types of infections. So that's why we need to take care of them. And also never mind about our homes. Here I specified augmented care units, for example. Not all the floors of the hospital have to be sampled, not all of them, because if others like the like other surgeries, simple surgeries like head and neck patients, those water outlets aren't sampled either because they are not at high risk. They won't get the infection, but if possible, I think we needed to, it would be good to have all the information and all the samplings, even in our homes, I think. Thank you. I think that was all for the questions. Thanks a lot for everyone.
Thank you all for coming. We have some refreshments over on that side, and you guys can feel free to eat, drink, and just have conversations.
Thank you very much. Ha, selfie mi? Olur. Olur. Olur mu? Tamam. If you don't mind, shall we take a photo all together? Senin telefonunu alayım Gözde. Teşekkür ederim. Good memory for us.
There is your sample stick.
Yeah, exactly. The only day I don't have it. Oh, I think I can get a